possible of challenge to drive novel techniques, forcing researchers to employ new ideas as well as reinvent old ideas to have fallen out of favor or become overshadowed by current buzzwords in the field. Some of the techniques that have been creatively harnessed to successfully shed light on the impact of microbial metabolism in atypical environments include microscopy and surface air interfaces, mass balance, radiolabeled fatty acid synthesis, and stereoisomeric ratios. The divergence between the rate of career span of the researcher of the rate of the subject matter is a central consideration in this context and can be overcome by intentional and rigorous long-term experimental design, mathematical modeling, teamwork, and global perspectives regarding measurement and impact. Microbial is a subject area within microbiology and genetic engineering. Microbial genetics studies microorganisms for different purposes. The microorganisms that are observed are bacteria and archaea. Some fungi and protozoa are also subjects used to study in this field. The studies of microorganisms involve studies of genotype and expression systems. Genotypes are the inherited compositions of an organism. Genetic engineering is a field of work and study within microbial genetics. The usage of recombinant DNA technology is a process of this work. The process involves creating recombinant DNA molecules through manipulating a DNA sequence. The DNA created is then in contact with a host organism. Cloning is also an example of genetic engineering. Since the discovery of microorganisms by Robert Hooke and Anthony van Leeuwenhoek during the period 1665 to 1885, they have been used to study many processes and have had applications in various areas of study in genetics. For example, microorganisms' rapid growth rates and short generation times are used by scientists to study evolution. Robert Hooke and Anthony van Leeuwenhoek discoveries involve depictions, observations, and descriptions of microorganisms. Mucor is the microfungus that Hooke presented and gave a depiction of his contribution, being Mucor as the first microorganism to be illustrated. Anthony van Leeuwenhoek's contribution to the microscopic protozoa and microscopic bacteria yielded to scientific observations and descriptions. These contributions were accomplished by a simple microscope, which led to the understanding of microbes today and continues the progress. Scientists understanding microbial genetics also has application in being able to study processes and pathways that are similar to those found in humans, such as drug metabolism. The role in understanding evolution. Microbial genetics can focus on Charles Darwin's work, and scientists have continued to study his work and theories by the use of microbes. Especially Darwin's theory of natural selection is a source used. Studying evolution by using microbial genetics involves scientists looking at evolutionary balance. An example of how they may accomplish this is studying natural selection of depth of microbes. Application of this knowledge comes from looking for the presence or absence in a variety of different ways. The ways include identifying certain pathways, genes, and functions. Once the subject is observed, scientists may compare it to a sequence of a conserved gene. The process of studying microbial evolution in this way lacks the ability to give a time scale and when the evolution took place. However, by testing evolution in this way, scientists can learn the rates and outcomes and evolution. Studying the relationship between microbes and the environment is a key component of microbial genetic evolution. Bacteria have been on this planet for approximately 3.5 billion years and are classified by their shape. Bacterial genetics studies the mechanisms of their heritable informations, their chromosomes, plasmids, transposons, and phages. Gene transfer systems that have been extensively studied in bacteria include genetic transformation, conjugation, and transduction. Natural transformation is a bacterial adaptation for DNA transfer between two cells through the intervening medium to uptake of donor DNA. It is combinational incorporation into the recipient chromosome depends on the expression of numerous bacterial genes whose products direct this process. In general, transformation is a complex energy requiring developmental process that appears to be an adaptation of requiring DNA damage. Fungi can be both multicellular and unicellular organisms and are distinguished from other microbes by the way they obtain nutrients. Fungi secrete enzymes into their surroundings to break down organic matter. Fungal genetics uses yeast and filamentous fungi as model organism of eukaryotic genetic research, including cell cycle regulation, chromatin structure, and gene regulation. Protozoa are unicellular organisms which have nuclei and ultramicroscopic cellular bodies within their cytoplasm. One particular aspect of protozoa that are of interest to human genetics are their flagella, which are very similar to human sperm spongula. Viruses are capsid encoding organisms composed of proteins and nucleic acids that can self-assemble after replication in a host cell using the host replication machinery. Are omnipresent in the biosphere, and their presence invariably affects the environment in which they grow. The effects of microbes on their environment can be beneficial or harmful or inapparent with regard to human measure or observation. The most significant effect of the microbes on Earth is their ability to recycle the primary elements that make up all living systems, especially carbon, oxygen, and nitrogen. The Earth is known as a co-system where materials cycle between lithosphere, rocks, atmosphere, air, hydrosphere, water, and biosphere organisms. Together, they make up all the components of our planet. Both living and non-living, Earth produces everything it needs to ensure the survival and growth of its residents. Environment is defined as the circumstances or conditions that surround an organism or group of organisms. Environment is a complex of social or cultural conditions that affect an individual or community. Since humans inhabit the natural world as well as the built of our technological, social, and cultural world, all constitute an important part of our environment. Environmental communities and their factors. The groups of similar species create population which results in a community. Microbial community means all microbial populations in a habitat. The activities of complex communities of microbes affect biogeochemical transformations in natural managed and engineered ecosystem. Microbial communities are very important for the rigorous process in the field of agriculture which increase the rate of crop production. Microbial community may be terrestrial or aquatic. Soil formation is a low process that involves physiochemical weathering and biological process over millions of years. Air, the atmosphere is an inhospitable climate for microbes because of the stress due to dehydration. This results in a limited time frame for microbes to be active. However, some microbes get resistance to these stresses through specific mechanisms promoting loss of their biological activity. Spore-forming bacteria, molds, fungi, and cyst-forming protozoans all have specific mechanisms through which they are protected from these harsh gaseous environments. Temperature is a critical factor influencing the activity of microbes. In general, high temperature leads to inactivation due to desiccation and protein denaturation, whereas lower temperature promotes longer survival rates. At a very low temperature, some microbes lose viability because of ice crystal formation on their surface due to freezing. Mostly radiation at low wavelength, e.g. UV radiation and ionic radiation X-rays, is harmful for microbes, causing DNA damage. The study of freshwater microenvironment is known as microlimnology. There are two types of freshwater environment: standing water in alinthic habitats and running water in lochtic habitats.